Thank you, Alex, for that kind of introduction. So today I'm excited to share kind of one project that we worked on jointly in the labs at Brunet and James at Stanford on decoding brain aging at spatial and single cell resolution using machine learning approaches. And so I'm very interested in the process of aging because I think of it as the greatest risk factor in many different types of chronic diseases. So these are diseases like cancer, heart failure, stroke, dementia, and diabetes. They're kind of very diverse diseases that target different organ systems, but they all share kind of ubiquitous risk factor, which is kind of this process that we know as aging. Aging is difficult to understand given its complexity. So to give you one example of this complexity, just at the level of organs where aging emerges, also travels at the level of different scales of biology. So in individual organs, they have complex spatial organization, tissues compose organs, tissues interact together to give rise to kind of organ level attributes. But even within tissues, there's individual cells and they interact to give rise to tissue level functions. So you can kind of appreciate how there's many layers of biology that are relevant to aging. And so a key question I was interested in addressing in my PhD was this question of whether we could use spatial transcriptomics, these newer technologies, we can measure, high dimensional gene expression in an individual cell, but also keep intact kind of the spatial or kind of hierarchical nature of the tissue to really understand cell-specific effects that might drive aging in an organ like the brain. And so to that end, I collected a single cell spatial transcriptomics data set of the adult mouse brain across life. This is tiling ages from 3.4 months to roughly 34.5 months. To give you a sense of the span of this age in terms of human equivalent years is roughly 20 years to roughly 95 years in terms of human age, and profiling roughly 20 ages across the entire kind of lifespan of the mouse. So in this case, we could use mirrorfish technology to really measure the expression of 300 genes across the entire brain sections, profiling both coronal sections and sagittal sections of the brain, and measuring these 300 genes and their expression at single cell resolution. And so in total, we generate roughly about 2 million high-quality cells across 18 unique cell types. So here I show on the left a UMAP representation where the 300-dimensional data set, so the 300 genes for each cell, is projected down to 2 dimensions for visualization. Each dot here is a cell, and it's colored by its cell type. But I think a more exciting way to visualize the data given its spatial information is to look at each cell in its native spatial context. So here visualized by the XY coordinates of each cell in kind of one representative coronal section of the mouse brain. And so you can appreciate here kind of spatial enrichment for different cell types, for instance, excitatory neurons in the cortex, inhibitory neurons in the striatum and adjacent regions of the brain. And then as a final example, oligodendrocytes enriched in the white matter tracts of the brain. And so far we generated this kind of large data set of brain aging at spatial and single cell resolution. Again, the motivation for this project was to ask the question whether we could track aging at single cell resolution and then build up a kind of systematic understanding of the spatial level using machine learning type approaches. And so towards that end, we built these models, which we call spatial aging clocks, that can measure aging at single cell resolution from spatial transcriptomics data. And so kind of in a nutshell, how this pipeline works is we take as input the spatial gene expression. So this is all the cells and the expression of the 300 genes in the mirrorfish data set. And then to enhance our signal over noise in these data sets, we designed Spatial Smooth, which is kind of a spatial preprocessing algorithm where we share information across graphs or networks of cells connected by spatial adjacency or proximity to each other. And then upon that processing, we were then able to train kind of cell type specific spatial aging clocks. So one aging clock for each cell type to predict age using very simple kind of machine learning. And so how well do these models work? They generally perform pretty well. So of the 18 independent models that we generated, about 14 have pretty high performance with a relatively high correlation between the predicted age, which I show here on the vertical axis, and then the actual age or the kind of the target label here on the horizontal axis. And you can appreciate that this is both true for kind of very common cells like excitatory neurons, medium spine neurons, astrocytes, and so forth, but also for very rare cell types like neurostem cells, macrophages, and T cells. And so far, I've shown that spatial aging clocks can predict age pretty accurately. I won't go into the exact details, but we've also validated that it generalizes well to other domains, so other brain regions, for instance, or across biological sex, and also that it generates pretty integrative kind of biomarker-like qualities in terms of predicting brain aging. So in the context of diseases like multiple sclerosis and Alzheimer's disease, we see accelerated aging reported by the models. In the case of rejuvenation, for instance, partial reprogramming or exercise, we see kind of rejuvenation or a lower predicted age under these models. So given these qualities, I was not so much interested just in using these aging clocks as ways of tracking aging, but really using them as tools for potentially discovering new biology. And so towards that end, I was interested in this question of whether some cells can influence aging of their neighbors in their local environment. And to address that question, I devised a kind of a matching approach here where you have defined an effector cell type. So these are kind of the circles here in pink, as exerting some influence on potential target cell type downstream, which is shown here in green. And to assess for the effect of the effector cell type on the target cell type, I can look at the predicted ages generated by these spatial aging clocks as a measure of the biological aging of these cells, either near effector cells paired or matched to target cells that are far from effector cells. And the controlling for confounders like, the brain region, the cell type, the age, and the sample, and so on. And so we can do this type of pair analysis for every combination of different effector cell types in the brain on every combination of different target cell types on the brain. And they can visualize each of these what we call cell proximity effects on aging as kind of the difference in the predicted ages in proximity or far from effector cell type for each of these interactions as a heat map. So here on the vertical axis is the effector cell type, which we believe exhibits or exerts some kind of cell proximity effect on the aging of the target cell type here on the horizontal axis. Everything here in red is basically a pro-aging effect, so a higher predicted age of the target cell when near the effector cell. Everything here in blue is basically a pro-rejuvenating effect, so a higher predicted age for the target cell when nearby the effector cell. And two kind of media trends were very striking to us, which are that T cells are very pro-aging across multiple cell types. You can see a lot of red across multiple cell types. And that secondly, that neurostem cells were the most pro-rejuvenating cell type by far across multiple different target cell types downstream. And so, of course, we've also looked at this through kind of more deep learning approaches. We can do kind of in silico perturbations, but what they point to kind of downstream is, the media question of where are the potential mediators of these cell proximity effects? And this is actually a difficult question to address using spatial transcriptomics, where you can only measure in the imaging-based approaches, like MERFISH at least, a limited panel of genes. So in case of the case of our study, we had 300 genes. There might be 500 genes in a different study, maybe 1,000 genes, but in either case, it's a far cry from the 20,000 or so genes that are known to be expressed at the whole transcriptome level. And so the idea here is that can we build new machine learning tools to really kind of bridge that gap? And so we leverage a tool that we built previously called Tissue, which leverages machine learning, particularly tools from conformal prediction to make that prediction of from 300 genes, can we predict or impute the missing expression up to 20,000 or so genes? And not only do that, but to do it in an uncertainty-aware fashion so we know what the error bars are for our predictions, and then do rigorous statistics to basically unbiasedly identify what potential mediating pathways might be behind the cell proximity effects that we identified. And so far, Tissue identifies these potential mediators of proximity effects. When we apply to our data set, we made the imputation 20,000 or so genes, unbiasedly looked at which genes increase or decrease along different pathways in the brain. For cells that are either near T cells, for instance, here in this case, we see that interferon response goes up in the cells near T cells, and then concomitantly in the T cells have the strongest pro-aging effect, there's actually elevated interferon production expression. At the same time, on the flip side of the coin, in neurostem cells, for the pro-rejuvenating effect, we see that downstream for the cells near neurostem cells, there's kind of altered lipid metabolism, and the most pro-rejuvenating neurostem cells have kind of elevated exosome processing pathways and also kind of elevation of growth factor secretion pathways. And so, of course, we wanted to perform orthogonal experiments, in this case, immunostaining, to confirm kind of these spatial associations that we predicted using our algorithms. And then one case, we show that basically the immunostaining confirms T cell mediators. And then the other case, looking at kind of immunostaining in the neurostem cells, we were able to examine that, for instance, in the neurostem cells, there's a kind of higher elevation of CD9 or exosome markers. And in the downstream cells nearby, these neurostem cells is kind of elevated to kind of fatty acid oxidation as predicted through our modeling approaches. And I think more strikingly that these two are also kind of linked spatially wise, so that the higher CD9 is also related to basically higher fatty acid oxidation in nearby cells. And so to kind of quickly summarize the project, we've generated the spatial transforms atlas of the aging brain across life, across multiple ages, and that gave us the ability to generate and train these spatial aging clocks, which are machine learning models to track aging at single cell resolution. And then from there, be able to use these tools to identify the spatial interactions to build up kind of one level of scale to understand pro-aging effective T cells and pro-rejuvenating effective neurostem cells, and then to bridge that to previous methods we developed to identify the potential mediating pathways underlying these proximity effects. So I want to give, quick thanks to my fantastic mentors, Brunet and James at Stanford, along with their respective labs and the fantastic collaborators who contributed to various parts of this project. And thank you again for having me. I'm happy to take any questions.